All right. So as a reminder, this Wednesday, we are having our test 2, and it is going to be just like test 1, same kind of like format, same idea where we'll just meet for an hour, and then the test will open at 9 a.m. and you'll have 1 hour to do it. And it'll be open from 9 to 11. So yeah, if you have any questions about the material on the test, please let me know, send me an email, visit me office hours, or if my office hours don't work for you, just schedule another time and we can meet then. That's perfectly fine. Yes, we still meet at 8 on Wednesday because since classes are 2 hours, we're going to spend the first hour going over some material for test 3, and then we will do test 2 right after that. Is the reading guide due on Wednesday still? Yes, we're still having that due on Wednesday for right now. So the person who sent me a direct message about the test, shoot me an email and we'll work it out from there instead of just like doing it in class. All right, so with that, let's get started. So just as a reminder, even though this part of the PowerPoint that we're on, chapter 7.2, it's in the test 2 folder, but it's not on test 2. So past here, not on test 2. Anything before this, when we were talking about hemoglobin, that is on test 2. Muscle contractions will show up on test 3. So just be aware of this. And I mentioned that last week, but yeah, anything we cover today, new material, won't see on the test. But let's get into how do your muscles work on a biochemical basis. And when talking about muscles, there are a lot of different structural details we can look at. So those of you who took A&P, you probably learned about the basic unit of the muscle, how it's made out of these repeating sarcomere units. And these sarcomeres have different bands, the I band, the A band, the H zone. They have different discs and all that. But what we're going to focus on in biochemistry is we're going to focus on the proteins that make muscle contractions happen. And there are 2 different proteins we're mainly going to focus on, and they're called thick filaments and thin filaments. And the actual name of these proteins is thick filaments are myosin, thin filaments are actin, right? And we're going to begin our discussion with myosin, shown here. And myosin's also shown here on the bottom as well, just different representations of the same protein. And in picture B on the bottom right of your screen, that's what myosin looks like in your muscles. So let's talk about this. So myosin is made out of 6 different peptide chains. Just as a reminder, a peptide chain is amino acids, end to C terminus. And we have 6 of these coming together to make one protein. So what we're looking at here is the quaternary structure of myosin. Remember, quaternary means many peptide chains coming together. And what is the 3D shape of those peptide chains? And we can break down these peptide chains into heavy and light chains. So you have 2 heavy chains and therefore 4 light chains. And these words heavy and light are referring to weight. And so if we go to the picture on the bottom left here, we can get a better idea of how these heavy and light chains interact. So the heavy chains, you got 2 of those, and they wrap around each other. So they're forming like a helix with each other. And at the very end, you have these head groups. So that's the N termini, the head groups. And where they end when they're wrapping, that's the C termini. So N terminus, C terminus. And wrapped around each head group are 2 of these light chains. So they bind at the N terminus of myosin. Now here, the C terminus, we have a pseudo repeat. And we talked about this when we looked at our fibrous proteins, where it's just a repeat of 7 amino acids, but it's called a pseudo repeat because it's not like the same 7 amino acids over and over and over again. Rather, it's the amino acids have the same properties, where it's either polar or nonpolar. And for myosin, position A and D, so the first amino acid and the 4th amino acid will always be nonpolar. And that's because that's where we're interacting, right? So for example, where the 2 chains overlap, right there could be the first position, and then right here could be the 4th position. We want these chains to interact, and the easiest way for that to happen inside the cell, just make them hydrophobic, and water will automatically stick them together. And inside our muscles, these separate myosin proteins are coming together to make this thick filament. And so these little bumps here are the head groups. And so you have one region where you have nothing but C terminal tail. So that's your bare zone. And then eventually you get to the myosin heads. And these are what are found in our muscles and help to make our muscles move. Now, first question here, just a little warm up, get your brains going on this Monday morning. Is myosin a globular or fibrous protein? And I have a poll for this, so let me just launch that. So which one do you think is myosin? Is it a globular or fibrous protein? We'll do about 20 seconds on this. So if you haven't answered, please do so now. Even if it's just a guess, please do answer. All right, let me end that, then download the results. Okay, downloaded. All right, so share those results so you can take a look at them. Most people answered both. And then we have fibrous next, one person with globular. I'm glad nobody selected neither. It is possible for it to be neither. Some proteins don't have shape, but myosin does have shape. And the correct answer here is both. This is a fibrous or fibrous and globular protein because the tail is reminiscent of a fibrous protein. And then the head group is reminiscent of a globular protein. So myosin has the structure components of both classes of proteins that we talked about earlier, both fibrous and globular. Now, myosin was our thick filament, and our next protein is actin, and actin is our thin filament. So this is the other protein that worked with myosin to make your muscles move. And actin has a lot of different jobs inside the cell, right? So when we talk about actin, we're not just talking about muscle. That's not the only place it's found. It's found in every one of your cells for the most part. And like it says right there, it's the most abundant cytosolic protein in eukaryotes. Actin makes up your cytoskeleton, right? It makes up the roads that other proteins inside your cells can travel. Here, we're not talking about that function of actin. We're mainly going to be focusing on actin's function in the muscle. And you have two forms. You have G-actin and F-actin. And this is the confusion I've seen every semester that I've taught this, but G-actin and F-actin, same protein, right? It's not like you have two different genes. You don't have like a G-actin gene or an F-actin gene. What the G and F stand for is just what state of actin that we're looking at. G-actin are monomers, where you just have actin by itself in a globular form. And this can come together. These can go and interact with each other to form a fiber that's called F-actin. And these can interconvert, right? F-actin can go back, they can break apart and go back to G-actin. G-actin can come together to form F-actin, right? Now, actin binds a lot of different molecules, ATP, calcium, magnesium. And when it's in its globular form, it's going to have ATP bound. Then when it becomes F-actin, it will go to ADP. And when we look at the structure of F-actin, it's a helix, a lot like DNA, and it does have orientation. It has a plus end and a minus end. And what those relate to is what part of the fiber is actin most likely to bind to. Let me just emphasize that, most likely. So you're most likely to bind at the plus end, and you're most likely to come off of the minus end, because you're just a fiber, so you can come off or go on either end. And I do want to stress it's either end. So even though we have a plus and minus end, it's possible for actin to add to the minus end, and it is possible for actin to leave from the plus end. But generally speaking, at the plus end, you have addition. At the minus end, you have subtraction, where actin comes on at the plus end and removed at the minus end. And the way I really like to think about actin, this is going to be an odd reference. I don't know if everybody will get it, but back in the day, when cell phones were starting to get really popular in the early 2000s, there was a game that was on every single cell phone called Snake. And you can still find it. And the idea of Snake is that you're a snake. And when your head moves, your tail follows you. And every time you eat something, your snake gets a little bit longer and a little bit longer. And in a sense, that's what actin is. It's like a rope. And if the rope moves, right, wherever the head goes, the tail will follow. but you can also make this rope bigger and smaller. When it comes to muscles, though, the actin isn't really going to be moving all that much. Inside the cell, it can, though. Inside the cell, and we're going to talk about this on a different slide, but inside your cells, not talk about the muscles, but other cells, actin is movable and the length can change a lot. When talking about muscles, though, we're going to be focusing on F-actin, because F-actin, the fibrous form of actin, is what is binding to our myosin, and that is what is making your muscles move. But before I go on talking about muscle contractions, any questions up to this point about either myosin or actin? All right. No questions? Let's move on then. So the last protein we're going to talk about that's important for muscle contractions is, well, it's actually two proteins, tropomyosin and troponin. And these proteins are interacting with the thin filament, which is actin. So remember, actin is thin, myosin is thick. And tropomyosin and troponin are kind of like a lock system, in that both tropomyosin and troponin bind to our actin. So if this is actin, on your actin, you're going to have this tropomyosin and troponin protein. So they're two proteins. So I'm going to draw them like kind of like that. And they act as locks. I guess it's more like a fiber than what I drew there, but the idea here is for your muscles to contract properly, actin needs to be naked. That is, actin needs to not have like any other protein on it. And once actin is naked, your myosin will go and grab it. And that starts your muscle contraction. However, when tropomyosin and troponin are present, they block myosin from binding actin, which is a good thing for the most part, because imagine you're sleeping. You don't want your muscles to start contracting. That's called a muscle cramp, and they hurt a lot. And you don't want that to happen if you don't want your muscles to move. So when you do want your muscles to move, you want tropomyosin and troponin to be removed from your actin. And the way that happens is through calcium. So troponin binds calcium. And when troponin binds calcium, so let's say calcium comes in, that means troponin will be removed. So troponin will bind the calcium and just fall off actin after it binds. Once troponin is removed, tropomyosin also falls off. So tropomyosin only interacts with actin if troponin is present. And troponin falls off if calcium is present. That's how that system works. So you need troponin to allow tropomyosin to stay on the actin. Calcium will kick troponin off, which is good. And imagine that you want your muscles to move, then you just send a nerve signal to your muscle. Your nerve will release calcium. That calcium binds troponin, and now you can have muscle movement. Right, so that's the regulation of our muscle movement, troponin and tropomyosin. So any questions about our regulation system there? All right. So again, another little simple poll just to make sure that we're all here, all paying attention. Let me launch this poll. Based on what I just said about 30 seconds ago, what statement is best to describe troponin? So what is that troponin again? Do about 30 more seconds. So please put in your answers by then. All right. 5, 4, 3, 2, 1. Download that. All right. So tropomyosin is a calcium binding, or sorry, troponin, not tropomyosin. Troponin is a calcium binding protein, right? As I just said, it's going to bind calcium. And then once it binds calcium, it falls off actin. Tropomyosin falls off actin as well. Now actin and myosin can interact and you can have muscle contractions. And that brings us to the diagram of how muscle contractions work. And this is called the power stroke. So let's start on this image 1. And here we have our thin and thick filaments. Now, we already have troponin and tropomyosin removed. So we have our naked actin, and the part of myosin that binds to actin is the myosin head group. So the N terminus is what is binding to our F actin here. So let's go through this process one by one. So step 1, ATP binds myosin. Once ATP binds myosin, myosin no longer binds actin. So myosin stops binding actin once ATP is bound to myosin. Once myosin is unbound from actin, it will hydrolyze ATP. So ATP at this step will go to ADP plus a phosphate. And breaking this ATP bond, as I'm sure you know from all your other biology classes, this bond is very high energy. And in proteins, one of the things they do when they break a high energy bond is that they use that energy to change their structure. They go through a rearrangement or a configuration. Here, what happens is that the myosin head actually moves forward when it breaks ATP, as shown in that image. That's what that arrow is showing, is that the myosin head moves forward. And once that ATP has been hydrolyzed, myosin is free to bind actin again. So it does it but it does it at a different position. If you go back to our first image, you can see that the actin we are originally binding to is orange. And after we've hydrolyzed, the myosin head we're binding to is in front of the orange one. So we have bound at a different actin monomer unit. Once we are bound, our phosphate is removed and we bind even stronger. So beforehand, we were binding only weakly. Phosphate leaves the myosin head group. Now we bind even stronger. And once that phosphate is gone, we do the power stroke. We go back to our original position. This has the effect of moving the thin filament back while the thick filament goes forward. So we are contracting our muscle when we do this. After the power stroke is done, we release ADP, and we can do this again. So that's how your muscles contract. And every year when I do this, I like to give a visual demonstration. So I'm going to stop the sharing so I can get on the camera here. So, all right. So the way that this works is oops, sorry, didn't mean to hit the mic there. So when thinking about this, even though we only consider one myosin head group, imagine there's like 1,000 copies of me, like all in a line. And my hands are going to represent the myosin head group, right? So let's go sideways like that. And I'm holding an actin. So imagine that in my hands, like there's a long cable that's running above me. I just have 1,000 copies of me holding this cable. And to begin with, I am bound to this cable. And then, let me see, do I have a marker here? I don't. Okay. So ATP binds. So I'm going to use my pen. So ATP binds. And when ATP binds, I no longer am bound to actin. Then I hydrolyze my ATP. And when I hydrolyze my ATP, I shoot forward. And once I hydrolyze that ATP, I can rebind. So I'm at a now forward position and I hang on. Then I release the phosphate and I hang on even tighter. Then I lose ADP. And when I do that, I go back to my original position, bringing the actin filament I'm hanging on to with me. And that's the power stroke. So bound, ATP, unbound. Hydrolyze ATP, shoot forward. Rebind, remove ADP, go back. Bound, dun, 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 dun. That's the power stroke. And that's how your muscles contract in that visual demonstration there. And let me reshare that. Get my Zoom back up. So any questions about the power stroke from this image or from my little demonstration there? All right. Well, if that is clear, then we can move on. And I have a question then for people. So when you die, your muscles become stiff. You are very hard to move a couple hours after you die. This is called rigor mortis. Based on what we just said, I want you to explain biochemically, molecularly, why does rigor mortis happen? Why do we become stiff when we die? based on what I just explained using the power stroke. When you have an idea, feel free to put that in chat. As always, you can send me a private message or make it public, either way. So the cells no longer have the energy to pump calcium out of the cell. So calcium concentration rises, forcing the muscles to remain in a contracted state. So that's part of it, right? So we no longer have the troponin, tropomyosin system working, right? If calcium's always available, then troponin won't bind to actin. And if troponin is not bound, then myosin's free to interact with actin. But there's one more part of it. Why, if I forcefully try to bend somebody's arm, that's going to be very hard to do. Our bodies no longer produce ATP. Yeah, that's the other part of it. So remember, based on the power stroke, myosin only releases actin if ATP is present. And if ATP is no longer being generated, there's no ATP to bind our myosin heads. So what happens is that once that actin is naked, myosin just grabs onto it. And if there's no ATP around, it won't let go. It just stays there. So all of your actin is going to be coated with myosin. Your muscles are not going to move anymore because the power stroke is gone. And so you would have to wait till the body decomposes further to get that movement again. Does it depend on temperature? So only when it comes probably to decomposition. I mean, if it's colder out, it'll probably just happen faster. And if it's hotter out, you'll probably be able to get past rigor mortis faster. But the process itself probably is not. So any questions about that or power stroke, actin, myosin, troponin, tropomyosin? Anything at all? All right. So if not, let's go to the next slide then. Could you explain again where troponin and tropomyosin are in the diagram? Sure. So thin filament, and then you have thick filament. So actin, myosin, tropomyosin. tropomyosin wraps around actin, so that's tropomyosin. And on this, you have troponin. If troponin and tropomyosin are on actin, then myosin cannot bind. However, if you have Ca come in, calcium, and that comes in, when that comes in, troponin binds it and is removed from actin. And once troponin is removed, tropomyosin is then removed as well. It just falls off. And once that falls off, then myosin is free to bind because you have naked actin now. And now you can do the power stroke. So troponin and tropomyosin are mainly regulation, and calcium controls that regulation function. Does that make sense? So it stays off as long as we're using that muscle. Yeah, as long as you still have calcium around, it's not going to rebind. Because once it's bound, you can no longer use that muscle. All right. So another question here that goes back to more or less general chemistry. When we're talking about the interactions of myosin to actin in the power stroke, are we breaking any covalent bonds in this cycle? And what type of interactions make myosin and actin interact? And I guess the question really that I want to ask, does actin and myosin make covalent bonds? So let's start with that question and just a simple yes or no. When actin and myosin interact with each other, do they make any covalent bonds? Yes or no? So feel free to put that in chat for me. Do you think when actin and myosin are interacting, are they making covalent bonds? What do people think? It's the interaction based on what we've talked about so far. Are actin and myosin making any covalent bonds between each other? Just a simple yes or no. That's all I'm looking for here. Yes, yes, no, yes, yes, no. All right, let me tally this up. And if you haven't answered yet, please just put yes or no. Doesn't matter if you're not sure. I'm just wondering where people are. So yes, 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 no, yes, no, yes, yes, yes. All right. So the correct answer is no. Right. So let's go back to the diagram. So if you remember, like a covalent bond is the sharing of electrons. And covalent bonds are very strong. Covalent bonds are kind of permanent when you make them for the most part. And actin and myosin, we're not making any covalent bonds here. This is all non-covalent interactions. And this is actually a big point in the broader sense of biochemistry as a field. When different proteins interact, they like never do so by covalent bonds. It's very rare that you're going to see two different proteins come together and make a covalent bond, because that's permanent. That's like super glue or gorilla glue, right? All of these interactions that we're talking about when different proteins, even when it came to hemoglobin, the different subunits, they don't make covalent bonds with each other. It's all non-covalent interactions. And that's what we're looking at here. So it's all non-covalent. So that's the first question I have. So the second question, are there any covalent bonds broken in the power stroke? And again, I'm just looking for a simple yes or no. So when we are doing the power stroke, are we breaking covalent bonds? Or are we making covalent bonds? So not talking about actin and myosin themselves, I'm talking about the whole system now. So I guess I can do also made. So again, just feel free to pop in a quick yes or no in the chat on what you think. Are we making or breaking any covalent bonds in the power stroke? Not sure, feel free to go back to that slide if you have your notes with you or on your computer. Refresh yourself with the power stroke. Good way to reinforce what I just said by asking yourself these types of questions and going back to refer at the model again. So I'm going to do the same type of tally once people are answering. So we have 1, da, da, da. So, wait, yes. So far we have what, 1, 2, 7 people answering. So those of you who have not answered, just feel free to put it in next 20 seconds. No with a question mark. All right. So the correct one here is yes. So actin and myosin don't make covalent bonds with each other. However, in the power stroke, we are breaking a covalent bond. The ATP is going to ADP in phosphate, right? So let me draw phosphate here really quick. Oops, it's P. And this is adenosine. So, oh, there's an O there. So ATP has 3 of these phosphates, And when you're breaking ATP, you just take one of these phosphates and you remove it. And that's what inorganic phosphate means. Right? Inorganic phosphate looks like this. So yes, we are breaking a phosphate covalent bond during the power stroke. Right. So any questions about thinking about the interactions of the power stroke? Or now that you had a couple minutes to stew on it, any questions about the power stroke itself? All right. So the last thing we're going to talk about is actin inside the cell, not in the muscles. Just briefly talk about it. Like I said, actin is their most abundant cytosolic protein. And what happens inside the cell is that you can get what is called treadmilling. So let's talk about treadmilling here briefly. Remember, you have two different forms of actin. You have your G-actin and you have your F-actin. Same protein, it's just, are you by yourself or are you in a fiber? And again, we have the plus end and the minus end. G-actin usually adds to the plus end, usually comes off at the minus end. It can happen either way, and that's what's being shown by these arrows. So at the plus end here, we have 6 actin coming on, but we actually have 1 actin leaving. At the minus end, we have 4 actin coming off, but 1 coming on. So you can add to either end, you can be removed from either end, but there is a preference. And for cells that want to move, the way they move, as shown in this picture, this cool image, is through their cytoskeleton, which is actin, right? And so if I want to move a part of my cell, I need to stretch actin this way. And the way that actin moves is through this treadmilling process. And this is really shown if you follow these green actins, in that you can see that they are moving through F-actin because you are continuously adding to one end and you're being removed from the other end. So the actin itself is traveling by adding to one end and removing by the other end. That process is treadmilling because it's a lot like a treadmill, where the thing that you run on the treadmill, it's continuously moving, but it's not like growing, right? And the actin itself, the fibrous actin is moving, but the whole length of it really doesn't grow because you have continuous addition and continuous subtraction. And those of you who go into microbiology or for some reason biochemistry, actin is talked about all the time. It's a control. Anytime you see like an experiment to see protein levels, like inside of a cell, the control they always show is actin to say, here's our protein, and let's compare it to the amount of actin inside the cell, because every cell has actin in it, and it's the most abundant one, so it's a good baseline. But yeah, that is our other use of actin that's not muscle related. And with that, that ends the PowerPoint from Wednesday. And let me get to the PowerPoint that I had created for today. During this transition, if you have any questions about anything, feel free to pop them in the chat. Otherwise, give me 1 minute while I switch PowerPoints here. share that. All right, so let's get to our PowerPoint for today. And we're just going to briefly talk about another protein system, the antibodies. And antibodies are part of your immune response. And I'm not going to really go over the immune system that much. Those of you who have taken classes in your biology courses probably know more about the immune system than I do, to be honest. But we're going to focus on one important protein of the immune system, and that's the antibody. So we're going to look at how an antibody binds an antigen. And an antigen is just something that an antibody binds to, and it's usually foreign. Either some kind of foreign protein or foreign sugar. It doesn't have to be foreign. Your antibodies can attach on something inside yourself that's part of you. That happens all the time. And it's not good when it happens. Some forms of arthritis are like that. But anything that triggers a response to an antibody, we call it an antigen. So what we're going to focus on in this class is really the structure of your antibody and how they bind to antigens. So let's look at the structure here on the left. And you have about 5 different types of antibodies inside your body. We're only going to focus on one. We're going to focus on the IgG, immunoglobulin variant G. And it has this Y shape, at least that's how I think of it. It looks like a letter Y. And there's 2 different regions of the antibody. You have the Fab region, which is like the claws or the tops of the Y. And you have the FC region, which is like the tail of the Y. And you can separate these two using a protein called papain. It just cuts right there. And then you get an FC and an FAB. And antibodies have 2 different chains. They have light chains, which I'll circle right here. So those are light chains. And then you have heavy chains. And this is just like myosin, in that the light and heavy is talking about weight. Light chains are lighter than heavy chains. So 2 heavy chains, 2 light chains. And you have 2 different regions as well that are denoted by V and C. So V stands for variable, C stands for constant. And the way that antibodies work is that these variable regions, these change. Every antibody has a different variable region. They have different amino acids that make up these regions. And because of that, your antibodies bind one antigen and one antigen only. So you have millions and millions of different possible antibodies you can make, but you're only going to bind to one antigen with that antibody. Then the FC region, that's the region that will communicate with the rest of your immune system. So when the variable region binds an antigen, this FC region will be sticking out. And this is what binds to your B cells to get the whole immuno response going. But for us, remember, heavy chain, light chain. remember variable region, constant region. And remember, the variable region is what is binding to our antigens. That's the main take home for us, the main things we're worried about. So let's get some biochemistry here. Using that image on the left, and I'm telling you that light chains are 23 kilodaltons, heavy chains are 65 kilodaltons. And remember, a kilodalton is just a unit of weight we use for biochemistry and chemistry. I want you to tell me, if I do an experiment with gel filtration, what would be the mass of my IgG? If I do SDS page, what would my gel look like with size? And if I use SDS page with 2-mercaptan ethanol, remember 2-mercaptan ethanol or beta-mercaptan ethanol, these break disulfide bonds. What would the gel look like there, and what sizes would you expect? So this is a review from topics we covered in chapter 1, or not chapter 1, but test 1, where we looked at these experimental techniques. So refresh yourself on those techniques and see if you can come up with the answers of these questions. What size would I expect my antibody to be through gel filtration? What would an SDS page look like? And what would an SDS page with 2-mercaptan ethanol look like? So I'm going to give people a few minutes to work on that. If you have questions about this, let me know. Otherwise, I will be back in just a few minutes to go over the answer with these. But please do try to do these on your own first. All right, so hopefully you made some progress on that. Let's take a look at this. So we have two light chains. So 2 times 23, oops, 23. So the weight of the light chains is 46 kilodaltons. We also have two heavy chains, each one 65. So 2 times 65 equals 130 kilodaltons. So the weight of one antibody is roughly 176 kilodaltons. Cell gel filtration, I would expect that if I did a gel filtration experiment, I would expect my antibody to be analyzed at a roughly 176 kilodaltons. Now let's do SDS page. So I'm going to do without beta mercaptoethanol and with beta mercaptoethanol. So that's what the plus and minus BME means. So the left-hand side is basically question B. The right-hand side is question C. So if I just do SDS page, I'll get one band. That's 176 kilodaltons. However, if I have beta mercaptoethanol, I'm going to break disulfide bonds. And so in this image, you can see the things that hold a light chain and a heavy chain together are disulfide bonds. That's what this SS is. So these are broken. So the light chains and heavy chains are no longer interacting. And the thing that holds the heavy chains together as well are disulfide bonds. So these are broken too. And so each heavy chain and each light chain are now separate from each other. So if I do SDS page with beta mercaptoethanol, I would expect two bands. One is my heavy chain band, which is roughly 46 kilodaltons. One is my light chain band, which is 23 kilodaltons. So disulfides are what are helping to hold our antibody together. And just remember, if you do SDS page and break disulfide bonds, then we would expect the chains to be independent of each other, and we could weigh them independently. But if we don't break these disulfide bonds, it just looks like one massive protein to us. So hopefully that was a good little refresher on how these experimental techniques work. Any questions about that? Would you not expect each light chain and heavy chain to move separately? They do, but they weigh the same, right? And since they weigh the same, they would appear at the same spot on your gel. So yes, I would expect two light chains here. I would expect two heavy chains here, but I can't distinguish them if they are the same weight, which is a drawback of SDS page, is that if you are trying to run multiple proteins, but they are the same exact weight, you can't distinguish them. They would appear as one band. I mean, the band might be like a little bit thicker than what I have here. Might not be nice and crisp. Might be like a fat band, but yeah, it would not be like two separate bands for a light chain or two separate bands for a heavy chain. No, it would be 23 and, oh yeah, 23 and 65. I put 46, my bad. I was looking at the wrong thing. It would be 23 and 65. You wouldn't add them together because they're separate, right? So when doing this, yeah, you have two light chains that are traveling. You got two heavy chains that are traveling through this gel. But since their weights are going to be exactly the same, they will stop at the same position. Yeah, I was looking at that math when I wrote 46. All right. So thinking about antibodies, antibodies have a very specific way they fold, in that you have two beta sheets forming together, held by a disulfide bond, and they make what's called a beta sandwich, which is exactly what it sounds like, two beta sheets just like folding together to make like a little sandwich. And this type of fold is called an immunoglobulin fold. So this type of fold is shown in all your antibodies. And if you ever find a protein with this type of fold, that's what the domain is called. This domain is called an immunoglobulin fold. And in this fold are the hypervariable loops. So that's what's being pointed here in these green. And these green parts of your protein, they don't really have structure, right? And they're variable. They can be like any amino acid. And they're found at the very tips of your antibodies. These are what are binding to your antigen. And it's all non-covalent, as we already talked about with the power stroke. Anytime you're making like interactions between proteins, it's almost always going to be non-covalent. So you're talking about hydrophobic, hydrogen bond, ionic interaction between your antibody and your antigen. And these interactions are extremely specific, and they bind very, very, very tight, where you have dissociation constants from -4 to -10 molar. Those numbers probably don't mean anything to you, but generally, once you get lower than 10 to the -6, that's very strong. And if you get like 10 to the -9 category, that's like super, super strong. Like you can't get much stronger interactions in biochemistry than that. And you see our antibodies can reach those quite easily. And the thing about antibodies, the IgGs, is that they are divalent. What I mean by divalent is if we look at our antibodies, they can bind two things at once. And so what happens inside your system is that if your body finds antigens, then your antibodies will bind, and it's going to form this giant lattice of antigen antibody. And you just get this huge pile of this antigen antibodies all building up. And this is one of the things that can signal the rest of your immune system to come over and start clearing this out. So white blood cells can come over and just start eating this whole mess for you. Your other antibodies are multivalent. So you have some antibodies that have 5 heads and can combine 5 things at once. And again, you're just making this whole lattice. But yeah, the important thing, again, to remember here are your variable and constant domains, your hypervariable loop. And for biochemistry, it's very important to realize antibodies only usually bind one thing. And we use that for experiments all the time. And we already mentioned that previously in this class. But for example, a Western blot. After you run a gel, you'll get these bands in the gel. However, you can't be certain that what's in that gel is a protein you're interested in. And so one technique to test that is called the Western blot. What happens with the Western blot is that you take the protein in the gel, and through electricity, you transfer the protein out of the gel and onto a membrane. And then basically what you do is you put an antibody on that membrane, and you then wash the antibodies off. And these antibodies are specific for your protein, right? So if antibodies stick to your Western blot, then that means your protein is there. If they don't, that means your protein isn't there. And this only works, this type of test only works because antibodies are so specific. They only bind one type of antigen. That's something very important to remember when it comes to biochemistry techniques. And anytime you see an antibody, realize they are incredibly specific. And so question 2, I actually already answered. So just for the sake of time, I'll just refresh yourself on that. So variation is only seen in the variable loops. Why don't we see it in the constant regions? So the variable loops have to bind almost anything, right? Anything that's foreign, they have to have the chance to bind it. And that means billions of different possibilities, billions different molecules that they could bind. So that's why we have variability here. However, the constant region of an antibody is what talks to the rest of your immune system. This is what is bound by beta cells. And so for our different antibodies, we always want our antibodies to talk to the rest of our immune system. Therefore, it makes sense that the tail will remain constant. Because if every antibody had a different tail, that means we would need to have a different protein to interact with those antibodies, which would be very wasteful in terms of energy. Because remember, we can make millions, billions of different antibodies. You don't want to make millions and billions of different proteins just to interact with those antibodies. So if every antibody has the same tail, well, we only need one protein to interact with all those different types of tails. And the only reason that we have variability at the tips is because you never know what you're going to encounter inside your cell. You have to be ready for the possibility of everything. All right, so any questions about our antibody structure or how it's interacting or anything we've talked about up to this point today? All right. So here's a good thought-provoking question. So if I have an antibody and I make it, and I have that antibody recognize a folded protein, why is it possible for that antibody not to bind the denatured protein? Let me draw a picture in case you're having a hard time imagining that. So I have a folded protein plus antibody. I have bind, and it binds. I have the same protein, but I unfold it. And I have the same antibody, but sometimes you get no binding. Why is that a possibility? Why, if I train my antibody to bind the folded protein, does it sometimes fail to recognize the unfolded protein? Anyone have any ideas about that phenomenon? This is a good review for what we talked about a week or two ago when it came to tertiary structure and protein folding. Because the shape of the protein changes? Yeah, that's not only the shape, but the characteristics of the protein changes. So let's imagine that our antibody binds to like a pocket that's like arginine, aspartic acid, and I don't know, cysteine. The protein is not in its native form, might not be functioning properly. That is true, but antibodies actually don't care if a protein is functioning or not. Antibodies look for specific shapes and specific characteristics. And by characteristics, I mean charge, polarity, or hydrophobic characteristics. So let's say our antibody binds to this pocket of the protein, and we're looking at the outside of the protein, where you have an arginine, which is positively charged, aspartic acid, which is negatively charged, and a cysteine. And so your antibody can recognize this and bind. And I'll be happy right there. So that's your antibody. Yay. However, this is the folded protein. And let's say arginine is residue 87, aspartic acid's residue 122, and cysteine's like residue 153, which happens in a folded protein all the time. You can bring amino acids that in the primary structure are far away from each other. But when it's folded, they become close. And so if this protein becomes unfolded, well, let me draw it like that. The arginine would be like right here at position 87. The aspartic acid would be over here at like 122. And the cysteine would be over here at 153. You no longer have this pocket. So if I'm the antibody and I'm looking for that pocket, I can't find it anymore because it's not there. It's unfolded. That's 153. So I no longer bind to that protein because I'm looking for a specific conformation of amino acids in a specific orientation. And then the unfolded structure, I lose that orientation. So that can happen with proteins sometimes. So it's very important when making your own antibodies to be aware of that and to test that out on your own system to make sure you won't have a problem like that. But any questions about that idea? All right, if not, let's go on. And like I mentioned earlier, when we first talked about antibodies, usually you want your antibodies to only bind foreign material, but they sometimes bind your own material, and that's autoimmune diseases. And these diseases aren't really, well, the whole way antibodies work in that you have tens of thousands of different molecules in your body. Yet our antibodies normally don't attack ourselves. And the mechanism for that isn't that well fleshed out. Generally, the B and T cells that make antibodies that attack ourselves, they are usually destroyed before they mature. And this is constantly happening inside your body, right? If you accidentally make an antibody that attacks your own system, normally your body can figure that out and stop that from happening. But not always. And so if you make an antibody that attacks yourself, you have just inherited an autoimmune disease. And a lot of times this happens, these show up after an injury or disease. And so here's a bunch of them. Arthritis probably being the most common and well-known, where your connective tissues are being attacked by your own antibodies and immune system, and you got that inflammation and pain. But type 1 diabetes is also that, where you have antibodies that attack your pancreatic beta cells and they destroy them. Your immune system destroys your own beta cells. Now you can no longer make insulin. So yeah, something that's not quite well understood. That's also an issue with organ or tissue transplants, where you get a new organ, a new tissue, and sometimes you'll make an antibody that sticks to that. It'll see it as foreign material, and it will attack that tissue and organ, which is a big problem when it comes to organ transplants. But yeah, those are autoimmune diseases. And with that, that ends chapter 7. We are, for the time being, until like chapter 10 or something, we are done with proteins. And we're going to move on to the next type of biomolecule inside the cell. So the next biomolecule we're going to look at are sugars. Whoopsies. Sugars. So remember, at the very beginning of this class, we talked about 4 major biomolecules. We have nucleotides, which we've already covered. We have amino acids, which we just finished. We have sugars, which are next. And after sugars, we have our lipids. So right now we're on the sugar chapter. And like nucleotides and amino acids, we're going to start small, looking at the monomers, and then go big, looking at our polysaccharides. And so to begin, there are a lot of different sugars. However, the only sugars that are really important in our body are the ones that I have boxed right here. So these are the ones I need you to know, both structure and name. And so on test 3, I'm definitely going to give you like a structure and say, what is the name of this sugar? Or I might give you a name and ask, what does the structure look like? But I'm only gonna ask those for certain sugars, and the sugars are boxed. Now, when it comes to sugars, you have 2 different forms. You have your aldoses, which are aldehydes, and your ketoses, which are ketones. And every sugar in the linear form has a C double bond O as part of the structure. If that C double bond O happens at position 1, it's an aldose. If it happens at position 2, it's a ketose. Right, so that's the difference between your aldoses and ketoses. What position is this carbonyl at? And we can also name, the other way we distinguish sugars is how many carbons they have. And if you only have 3 sugars, you're called a triose. 4 sugars, yeah, you're a tetrose. 5, you're a pentose. 6, you're a heptose. And you can keep going on. Heptose, octose. I don't know what a 9 would be, like a nonose. Decose. But yeah, that's the general way you classify sugars. 1, you classify them by aldose or ketose. Then 2, you classify them based on how many carbons make up your sugar. So right now, all we're looking at is sugars in their linear forms. So make sure you know the box sugars for test 3. And the most important sugar inside the body, inside the cells, is glucose. Glucose is the main energy of the cell. That's how we make ATP, is through the breakdown of glucose. So let's look at glucose's structure. And glucose has 4 different chiral carbons. Remember, in our brief discussion of chirality, a chiral carbon has 4 different things attached to it. So 2, 3, 4, and 5 all have 4 different molecules attached to it. Carbon 1 is not chiral because it only has 3 atoms attached to it. Carbon 6 is not chiral because it has 2 of the same atom attached to it. It has two hydrogens, so that does not make a chiral. And when it comes to sugars, we have what are called stereoisomers. What a stereoisomer is it's a sugar that has the same number of carbons, but the chirality of the carbons is different. And the quick math for this is you do 2 to the power of how many chiral carbons you have. This 2 is because there's only 2 directions. You can either have the OH on the right or the OH on the left. So that's what the 2 means. And just raise it to the power of number of chiral carbons. So glucose has 16 different stereoisomers. You can make 16 different forms of this sugar based on how you do the chirality of these carbons. Inside the cell, most sugars we deal with are D. And the way you tell D versus L in sugars is that you look at the chiral carbon furthest away from the carbonyl. For glucose, that's carbon 5. If the OH is on the right, that's called a D sugar. If the OH is on the left, that's called an L sugar. So here we are looking at D-glucose. And the last terminology that sugars have are what are called epimers. An epimer is a special stereoisomer. And that is only 1 chiral carbon is different. So epimers are usually pairs. So here I have the epimer of glucose at C2 is mannose. So mannose looks like what I'm drawing here. That's D-mannose, where only carbon 2 is different. So these are stereoisomers of each other. And they are also epimers of each other. So every epimer is a stereoisomer. Not every stereoisomer is an epimer. That's the classification. Epimer is more specific than stereoisomer. So before we move on, any questions about that type of terminology? All right. So let's test that. I want to know for a ketopentose, a ketohexose, and a ketoheptose, how many stereoisomers are possible? I'll do A for us. So ketopentose, let's look at this name. So keto means we're dealing with a ketone. That means our carbonyl will be at carbon 2. And a pentose means we have 5 sugars. So let's draw this. Sure, I'll draw it this way. So this is a ketopentose. And we have 1, 2 chiral carbons. Positions 1, 2, and 5 are not chiral. So since we only have 2 positions, to find the number of stereoisomers, it's 2 raised to the power of chiral carbons. So we only have 2 possibilities here. So there are 4 different ketopentoses I could draw. So I drew one version where both OHs are on the right. Another version, is this. So here's one stereoisomer. Here's the second stereoisomer. And I'm not drawing the rest of this, just the chiral carbons to make this faster. Version 3 is if they're both on the left. And version 4 would be if this is on the right, but this is on the left. So those are the 4 different ketopentoses I can have. Again, the math is relatively easy here, 2 to the power of chiral carbons. So just to make sure you understand that idea, take a minute or so and tell me for ketohexoses, 6, or ketoheptoses, 7, how many stereoisomers are possible. So see if you can figure that out, and I'll be back in a minute or two to discuss this. If you have questions, feel free to put them in chat. All right, so hopefully you could wrap your head around that, or it's got part of the way. So let's look at a ketohexose. So 1, 2, 3, 4, 5, 6. We have 6 carbons. It's a ketone, so the carbonyl goes on the second. H2OH at the top, H2OH at the bottom. That means 1 chiral carbon, 2 chiral carbons, 3 chiral carbons. So we have 3 different chiral carbons. So this is 2 raised to the power of 3, which is 8 possibilities. And a ketoheptose would have 1, 2, 3, 4, 5, 6, 7 carbons. Ketone, second carbon, carbonyl, carbon 1, not chiral, carbon 7, not chiral. So we have 1, 2, 3, 4 chiral carbons. Our mass is 2 raised to the power of chiral carbon. So 2 raised to the power of 4. 16 different possibilities with the ketoheptose. 16 different stereoisomers. Right. Any questions about stereoisomers, sugars, or anything up to this point? All right. Question 5 deals with epimers. This is true or false. So I have a poll for this. Remember, an epimer is only different at 1 chiral carbon. So see if you can figure out these true and falses. I'm gonna put up the poll. Feel free to minimize the poll if it's getting in your way and then come back to answer it. But just let me know. Of all those possibilities I put up there, true or false, are they epimers? So true would mean they are epimers. False would mean they are not epimers. So take a few minutes, see if you understand epimers and that you can get these. Again, I'll be back to discuss. If you have questions, let me know. And we'll do about 1 more minute of this or so, then get to discussion. So please be sure to answer in the next minute. Even if it's just not sure about the other ones, just put in guesses if you're not sure. Okay, 15 seconds. So please answer if you have not done so. All right. That's that. Let's take a look at these. So sorbose and psychose. So sorbose is right here, psychose is right here, and they only differ in this carbon. So that is true. They are epimers. How about sorbose and fructose then? So sorbose and fructose are right next to each other. They differ at this carbon and this carbon. So that's more than one. They're not epimers. Next, we have erythrose and arabinose. Arabinose is right here, and etherose is right here. So they cannot be epimers because they have different number of carbons. So that is impossible. Then we have D-fructose and L-fructose. This is the trickiest one. So D-fructose is here. And let me draw L-fructose. So we have 6 carbons. So this is tricky because I said that the only difference between a D and L is the last carbon, or the last chiral carbon. What direction are you pointing? If you're pointing to the right, you're D. If you're pointing to the left, you're L. So that's L-fructose. However, the one thing I didn't mention is that if I switch, well, let me take a step back. The thing that makes this fructose is that the last chiral carbon is pointing this direction. The one above it is pointing in the same direction. And the chiral carbon is pointing in the opposite direction. That's fructose. So if I'm L-fructose, I have to follow that. So the chiral carbon right above it has to point in the same direction. And then this one has to point in the opposite direction. Then we have our carbonyl. So that's L-fructose. So they differ in every single chiral carbon. So that is false. They are not epimers. So yeah, just keep that in mind. Although D and L are distinguished by the last chiral carbon, what direction are you pointing? What makes a sugar a specific sugar is the relationship to the other chiral carbons to that last carbon. So here we have left, right, right. Therefore, L-fructose would be right, left, left. All right, so let's finish this one off. Arabinose and ribose, we are differing at the second carbon only, so that is an epimer. Ribose and ribulose, we're looking at an aldehyde and a ketone, therefore, by definition, they cannot be epimers, so that's also false. And that is where we're going to end our discussion for today. Any questions people might have on anything here in the last moment? Or you can also catch me in my office hours today or send me an email. But anything people have that they just want to pop in chat while I close all of this down. All right, since I'm not seeing anything, that's all I have for you. I'll put a homework up as always. And let me know if you have any questions about anything before I test on Wednesday. Otherwise, see you on Wednesday. Study hard and have a good rest of your day.